My name is Thomas Holm Pedersen. I'm CEO of NMD Pharma. I'm actually also a founder of the company, and I guess I was also part of the scientific inventor behind this discovery that we are now pursuing forward as a treatment option. So because of that, I felt it was natural to perhaps go back a little bit in time and explain you a little bit about who we are and where we're coming from. NMD Pharma is a Danish company. We are located in the second biggest city in Denmark. It's called Aarhus, and the reason we're there is the underlying scientific discoveries that led to NMD were made at a university, Aarhus University, that you can see indicated there on the map here over on the right, where you have an indicator of where Aarhus actually is. The picture you're seeing in front of you is basically from a Christmas lunch in a very small laboratory at Aarhus University back in 2006. That was called the Muscle Lab back in the day, and it was a laboratory where I personally did my PhD. You can see me there in the back. And it was a muscle laboratory that built on more than 50 years of academic research within neuromuscular disease and especially ion channel biophysics, so special type of proteins that are expressed in many different tissues. But we were very interested in muscle biology and the basics of how electrical signals are transmitted from the brain to muscle fibers. And I did my PhD there. I did my PhD and was very interested in a particular ion channel called CLC1. And the discoveries that we made in that laboratory back in 2006, entirely academic, led to, let's say, the foundation for actually starting the company and to the very idea of using CLC1 as a new approach for targeting skeletal muscle. This is from 2006, as you can see. I just handed in my PhD at that stage, and this was actually part of my PhD program to propose to use CLC1 as that target. A lot has happened since then, and if you move one forward, we spun out from the university at Aarhus. We and formed a company. This is something we did in 2015. The company was created, and today we are 53 people in the company. We are located with the headquarters of the company in Aarhus, still in Denmark. There that we just showed you on the map. We also have an office in Copenhagen. We have an office in Boston as well, and we also have staff who live in both France and also in Spain and the UK. So we've certainly grown a lot, but we are still a small company, but with a lot of really great experts within all areas of drug development, especially focusing in on neuromuscular diseases. And our company is small, but we have a very, very clear focus to develop novel therapeutics for neuromuscular diseases. And so I think if we sort of try to capture exactly what we're doing, if you move one on, Thomas, we are a company that has a very clear vision. We really hope that we can contribute to creating a future in which patients can, with neuromuscular diseases, can live better and more independent lives. We think it is key, and that's the mission. We think it is key in order to achieve that we can find new therapies that can really improve on our skeletal muscle function, that can improve our strength, mobility, and so that patients actually have the opportunity to actually become more mobile and actually are better able to do very important everyday activities that can be challenging if you do not have the appropriate skeletal muscle strength to do that. So that's really what we hope to do. And the way we are doing that, I'd like to, first of all, tell you a little bit about what exactly we are pursuing with a compound called NMD670, a little bit about how this mechanism actually works, so you have a good sense of what exactly we're trying to do. And then what's going to happen then is Thomas will tell you a little bit more specifically about the ongoing clinical trial. So what we are specifically developing is we're developing molecules, small molecules that can actually be fitted into a tablet, so you will be able to oral administration of this small molecule. And these small molecules, what they do is that they inhibit a particular ion channel, and this is the ion channel that was also the interest of our research back in the day in the academic setting, this CLC1 chloride ion channel. Now, what is important about this particular ion channel is that it is only expressed in skeletal muscle, so it is not expressed anywhere else. And when you are developing new therapeutics, you really want to have them as specific as you possibly can. And a way to have these compounds therapies as specific as you possibly can, of course, is that you carefully select your target and the thing you want to really change in the biology to be as selective and relevant for the particular function you want to improve on. And then we're really lucky with CLC1 because it is very selective for the relevant tissue that is we are trying to improve function of. The way we are doing it is through a compound called NMD670, and this is a small chemical that actually inhibits CLC1. The way it is administered is through a tablet that you take twice a day, and you take them 5 to 8 hours apart. And with that, we predict that you would have effect of these therapy throughout the awakening time of the day and into the evening as well. That is really what we predict with this. And what it does is, and I'll show you a little bit more on the next slide, but we'll not go there yet. But on the next slide, we will show you how exactly this mechanism actually works. In short, what it does is it improves how the nerves are connecting with the skeletal muscle, how that signal that is in the nerve is actually transmitted to a signal also in the muscle fiber. And this signal has to go from the nerve into the muscle fiber in order for the muscle fibers to be told to contract, basically. So if the signal cannot get from the nerve into the muscle fiber is not activated and therefore it doesn't contract. And I'll show you later, we know that in Charcot-Marie-Tooth disease, there is a failure in this connection point between the nerve and the muscle. And the CLC1 inhibitors, we hope, and we have evidence to show that, can actually improve the function of this connection point. And by bringing back the ability of the nerve to activate the muscle fibers, we believe that we can enhance muscle strength, the power, and also endurance when you are doing more, let's say, prolonged activities. That would be hopefully addressing some important uncovered need and symptoms in Charcot-Marie-Tooth disease. So NMD670 is the most advanced of our compound. We have several different small molecules that can inhibit CLC1. You can see a pipeline here on the left where NMD670 is currently being tested in phase 2 studies in both myasthenia gravis, in spinal muscular atrophy, and in Charcot-Marie-Tooth disease. We already have actually tested NMD670 in a small myasthenia gravis trial, where we were able to see that NMD670 improved the hand grip strength. It was able to improve actually on some bulbar functions, including actually speaking. And also in this particular disease, it is very important to improve eye function. We could see that NMD670 improved all of these functions in myasthenia patients. And now we, of course, want to also move this forward also for Charcot-Marie-Tooth disease. So a little bit more about the mechanism. The diagram here on the left shows a very simplified version of how we activate our muscle fibers. As you know, as we are moving our muscle and moving our arms and legs, and when we walk and so on, we do that very deliberate. It's voluntary contractions. That means a signal arises in the brain. It then travels down the spinal cord, and now imagine the signal to be this blue line here, blue, almost like lightning ball, that travels down from the brain down through the spinal cord, and then it travels further along from the spinal cord out and sort of approaches the muscle fiber. And the nerve is then connected to the muscle fiber, and the electrical signal in the nerve, the blue lightning, has to go from the nerve into the muscle. And if it cannot be transmitted from the nerve into the muscle fibers will not activate. This very important transition point from the nerve to the muscle fiber is what we call the neuromuscular synapse, and that is what is enlarged there in this in the circle here. And I'm not going to go into a lot of details in this, but this is just to highlight there is a a well-described number of processes happening here at the neuromuscular junction, where there are certain substances that are transmitted, and there are some ions that are moving. So this would be sodium and chloride and potassium ions that are moving from the outside to the inside of the muscle fibers. And an important ion that is moving is also chloride. And this is the red negative sign of chloride moving from the outside and in. And when this chloride is moving in through CLC1, it actually counteracts that the muscle fibers are activated. And when you have this counteracting effect of CLC1, it means it actually opposes that muscle fibers are activated. Now, if you put that in a situation of a neuromuscular disease where the motor nerve is not working ideally, or even if the muscle fibers is not well tuned to react to what the nerve is signaling, then this inhibition of CLC1 becomes far too strong. So what we understood and what we developed scientific basis for is that if we can inhibit CLC1, we can take away some of this inhibition of CLC1. So there will be less chloride ions that flow into the muscle fiber, and thereby it becomes easier to activate the muscle fibers. And that is a way of, let's say, mitigating that the motor nerve is not working ideally and allowing that muscle fibers can be activated despite that the motor nerve is not optimally working. So it is a way of re-enabling the nerve to activate the muscle fibers. And our NMD670 is represented here by the red circle of NMD compound or CMPD. That means this is NMD670. What it does is it blocks this channel here in the interface between the nerve and the muscle fiber. And that actually enables the muscle fiber to better be activated. So this exactly is how it works. Now, a nice aspect of this and an important aspect of this mechanism is that it could potentially be used and could potentially benefit a number of different groups of people living with neuromuscular diseases. We already know that we have seen benefits of this in myasthenia gravis. So of course we are therefore also aiming to develop this also for myasthenia. Hence, we're doing a myasthenia phase 2 study. But we also have very strong scientific rationale for progressing this also in Charcot-Marie-Tooth disease. And Thomas, if you would move on to the next one. The reason we have that scientific foundation, if you click one forward, is that we actually conducted a study in Charcot-Marie-Tooth type 1 and type 2 patients, where we started out using clinical examination of how well is this junction or this connection point between the nerve and the muscle fiber actually working in patients with Charcot-Marie-Tooth disease type 1 and type 2. And what we could see is that in CMT patients, there is a clear deficit between the nerve and the muscle. So the function of that connection point between the nerve and the muscle is not optimally working in CMT, and it is an important part of the disease. It was somewhat unknown before we made this discovery, but we could now see that there is a dysfunction at this interface between the nerve and the muscle. And having that insight, we started to explore, well, if there is this insight, how well does it actually then relate to the symptoms? And what we could see, and that's illustrated in the middle panel here, is the worse nerve-muscle communication you have, the worse muscle symptoms you had, or vice versa. The more severe muscle symptoms we could observe, and this could be challenges with balance, it could be challenges with strength in the lower legs, it could be challenges with walking and walking speed. The more severe these symptoms were, the more pronounced were the defect at the interface between the nerve and the muscle fiber. So we now had observed that there is defect at the nerve-muscle junction, and the severity of that was a very strong predictor of the symptom severity as well. And this is the type of deficit that we believe our NMD670 can correct. To first test that even further, we also evaluated this treatment approach in animal models of Charcot-Marie-Tooth disease. This is what you see over in the third panel, where we actually have explored both type 1 and type 2 animal models. And I'm not going to go through a lot of detail here, but suffice to say that the size of the bars is showing how much force you could recover with our NMD670 compound in an animal model of CMT. In this case, CMT type 2D, but we have similar data also for animal models of CMT1A, where we can recover the contractile force. The blue bars are very large, showing really that you have a recovery of muscle strength in the animals, whereas if it's a wild type animal, that is of course a healthy animal, we are not seeing any improvement. So in the setting of disease, we're actually seeing a recovery. I think I can sum this down before I hand over to Thomas to speak about what we're doing in the future, is to say that we have discovered that in Charcot-Marie-Tooth disease, there is a clear, and this is actually in human, we've seen this in patients, that there is a clear deficit in the interface between the motor nerve and the muscle fiber. This is something that we have also discovered a new way to improve on, and we have with the CLC1 inhibition, and we have confirmed in animal models that this approach of CLC1 inhibition can restore muscle strength in the animal models. So we are now conducting a phase 2 study, hoping to demonstrate for the first time that CLC1 inhibition with NMD670 would be a new way to improve muscle strength, power, and endurance. So that is really the aim of the ongoing phase 2 study. And the ongoing phase 2 study is the first time we're testing this and exploring this in CMT. We really hope we can see these benefits of new therapies, and as we hope this going forward, and we certainly believe it's going to show this as we go forward, we would have to do subsequent larger studies, perhaps, to say that this is the first study, but there will be another study subsequent to that as well. So I think I'm going to stop here, hand it over to the other Thomas to explain exactly what we're doing in our clinical trial. So over to you, Thomas.
Thank you so much, Thomas. And also before I kick off here, a big thanks to HNF and to all of you for participating. I'm very excited to now tell you more about the study that we are doing at the moment. This is, as Thomas said, a phase 2A clinical trial where we, for the very first time, actually expose participants with CMT to this new molecule. The fact that this is a phase 2A study, that means that the study is the first time that we expose patients to this drug, or patients with CMT in particular. And the main objective of this type of trial, it is to investigate in a relatively small population of patients whether the drug actually works as we hope it will, and also whether the beneficial safety profile that we observed when we studied the drug in healthy participants during what is called a phase 1 study can actually be repeated in the patient population for which the drug is intended. If this study, the ongoing study here, is successful, as Thomas said, the next step would be a larger study, what is called a phase 3 study, where we would then be confirming what we hope to see in phase 2. If that study is then successful, this will allow us to submit an application to market the drug, and only at that stage can the drug actually be marketed and sold. In this particular phase 2A study, we will include a total of 80 adult participants with CMT types 1 and 2, and these will be randomized to receive either NMD670 or a placebo treatment, which is a treatment without any active drug. And this is to really test and to make sure that the treatment effects that we hope to see, they are actually genuine treatment effects. And this is why we are including a placebo arm here. All the participants that will be randomized in this study, they will receive either placebo or NMD670 for a total of 21 days. The treatment will be received twice daily, and before, during, and after the treatment period is completed, we will conduct a range of different tests to ascertain whether the symptoms of weakness and fatigue are alleviated. That is to assess whether the drug it works, but also to test the safety of the drug. We plan in this particular study to include approximately 30% of the population with CMT type 2. And the reason for this is that this will allow us to understand if the drug works equally well, better, or not so well in this subpopulation of CMT. In this study, the primary endpoint, meaning the primary test that will be used for the study, it's called the 6-minute walk test. So this is a test where participants will be asked to walk back and forth in a corridor, and we will assess how long the participant can walk over 6 minutes. And this test, it assesses muscle mobility, fatigue, and it actually also requires well-functioning strength. But we include a range of other efficacy endpoints in this study with the goal of understanding what endpoint is the right endpoint for our mechanism, so that we can properly design the next study, the phase 3 study. Some key facts about this study should you consider to participate. The study duration is approximately 8 weeks, and it includes 6 on-site visits. If you are enrolled into this study, you will take NMD670 or placebo twice daily for 21 days. There'll be 50% chance that you receive NMD670 and 50% chance that you receive placebo, and you will not know what you received until after the study is finalized. During the study, the participant will continue to receive his or her normal care, and that means medication to alleviate neuropathic pain and physical therapy. In all clinical trials, there are inclusion and exclusion criteria for participation. And in a phase 2A study, you normally have quite a homogeneous population to maximize the likelihood of actually seeing positive signals. In this study, participants, they need to be between 18 and 70 years of age. They also need to have a genetic confirmation of CMT type 1 or CMT type 2. This also means that participants with CMT type X or CMT type 4 are not permitted into this initial study. We do have reason to believe that the drug also works in these other subpopulations, but to maximize the likelihood of success in this initial study, we've confined the study to CMT type 1 and CMT type 2, because these were the patients that were studied during the observational study that Thomas also talked to before. There are some other disease characteristics that are required for participation in this study. One is that participants need to be able to walk within 100 meters of what can be considered normal in an age-adjusted CMT population. And this is assessed on the primary endpoint, which is the 6-minute walk test. Again, this is done to maximize the likelihood of actually seeing a signal in this initial study. We are also only including participants with mild to severe ankle weakness, and this is to ensure that there is actual residual symptoms in the patients that are participating, which will enable us to improve on some of the endpoints that we are testing in the study. There are other requirements for participations that includes, but is not restricted to, BMI, where we have an allowed range of 18 to 35 and a minimal weight of 40 kilos. Because this is an early phase study, and to ensure maximum safety of the participants, there are also requirements for contraceptive use by men and women in this study. There are also some key exclusion criteria for the study. One is that participants cannot have other significant disease that could interfere with the objectives of the trial. We also do not allow participants in this initial study with severe deformity or ankle contractures. And also, we are not permitting the daily use of assistive devices such as ankle-foot orthosis. Again, this is not because we don't think that the drug will not work in those participants that are using AFOs. It is to create a homogeneous population and thereby maximize the chances of actually establishing a positive signal. Then we also do not allow female participants who are pregnant or female participants who plan to become pregnant or are currently breastfeeding during the study. Again, because this is an early phase study and we want to ensure maximum safety at this stage. There is a total of 19 trial sites participating in this study. These are spread across Europe and perhaps more relevant to this particular webinar, also across the US. In the US, we have trial sites in Boston, Rochester, Kansas City, New York, Columbus, Columbia, Spokane, and Austin. And all of these trial sites are actually recruiting at the moment. If you are interested in participating in this study, or if you're just interested to know more about the study, then there is a website called clinicaltrials.gov, which have more information about the study. And you can find that web page by scanning the QR code here or by taking the NCT number, which is also included on this slide, and then putting that into the search field of the database. That web page will also have information about contact details for the individual sites, and in particular the site coordinators that are responsible for then arranging a screening visit, which is the first step towards participation, where the trial site together with you evaluate whether you can and whether you want to participate in the study. You're also more than welcome to reach out directly to NMD Pharma. We have a contact email address on our webpage where we will aim to answer any question of more general nature. But I do really encourage you, if you are interested in participating in the trial, to go to this webpage and find contact information for the site. Otherwise, we are happy to provide you with that contact information, and you can contact myself on this email address, or find our webpage and then our contact email address. I think I will pause there and extend a big acknowledgement and thank you to all the individuals and families who participated in the studies that we presented here today, and as well the ongoing study. And of course also to the investigators and site staff for performing the studies. And again, a big thanks to all of you. And then we are happy to take questions.
Thank you so much to both of you. It looks like we already have some questions in the chat and the Q&A. What is the age limit for participants?
Sure. So in this initial study, patients are required to be between 18 and up to 70 years of age.
Great. And I know this is based on the success of the trial, but is there an estimate? Pragna wants to know if there's an estimated time on when this could potentially be available on the market, if all goes well.
Yeah, so we are expecting that if everything goes according to plan, we would be submitting a submission for marketing authorization in 29. Exactly when that will happen, I mean, this is, but obviously we'll have to see as we progress forward. But that would be that's our hope. Yeah.
Would the muscle strengthening effects, in theory, potentially reverse the drop foot caused by CMT? Is that what you're hoping to see, or more of a stabilization?
That's a very good question, and I think the real answer to that is we are not able to answer it very specifically right now. What we know that our mechanism, what we believe our mechanism can do, is it can enhance muscle strength pretty fast, in fact. And you would also see that the 21 days is a rather short study to show that efficacy. Whether we then have sort of a secondary longer term effect, we may have reason to believe that, but let's see. One of the aspects we think that it would provide is indeed a better muscle strength, and so it is certainly conceivable that sort of things like drop foot would actually be possible to improve on. Anything you have on that as well, Thomas? Any reflection from your side on this?
No, only to say that this is, of course, something that we'll be studying both within the current study and in potential subsequent studies. So, yeah.
Thomas, we had a request if you could go back to the slide with all of the clinical trial sites and the contact info. Perfect. Thank you. Someone was asking for that. As far as those individuals who don't have a confirmatory genetic diagnosis, but perhaps their doctor said this looks like it could potentially be a CMT2, will they, you're still requiring genetic confirmation to be part of the trial, but could this drug perhaps be beneficial to many more types of CMT? I know you mentioned CMTX, but do you see it being potentially, available for CMT types 4 or down the line to a wider spectrum of subtypes?
That's definitely our ambition to expand to, and I think that's one of the important features of this particular type of mechanism that we are developing here, is that we believe it will be applicable across subtypes of CMT as well as across different diseases. So in that way, we think it's going to be hopefully reaching a lot of patients with similar challenges with muscle weakness and fatigue.
Very exciting.
And on your specific questions about confirmatory genetic testing, should there be a participant without genetic confirmation of disease, then it is also possible within the clinical trial protocol to actually have genetic testing, and that is something that the participant can discuss with the individual trial sites if this can be done or not.
Great. And are there plans to expand the studies to pediatrics?
Yes, there is. Not this particular study, but there's plans to expand the mechanism over time to also reach pediatric. When you are developing a new therapy for pediatric, you have to take several different steps along the way, including also qualifying it for use in pediatric populations, even in studies that are not conducted in the clinical, even some other types of parallel studies you need to do. These have not all been conducted yet, and hence we are currently limited to working with adults. But over time, we do realize there's a big need there as well in the pediatric population. And frankly, we have to develop that also, because there are also regulations that require us to have that plan for going forward as well.
Any plans to expand the clinical trials to Europe at all?
It is being conducted in
that's-
Europe as well. Yeah.
Okay. Yeah, we had someone.
It is, and there is also a slide within this deck. I think it is hidden, but allow me to just go out of slide mode real quickly and then put it up again.
Oh.
So there is a number of trial sites across different European countries. So we have in this study trial sites in Belgium, in Denmark, in France, and in Spain. So certainly there's possibilities in Europe as well, and all these trial sites they are also recruiting now.
Great. And I know you spoke about potential or ruling out potential side effects and testing for the safety of this drug. Have you observed any side effects so far in your preliminary studies, and what were those potential side effects?
Yeah, so this particular mechanism, we actually know a lot about what we could anticipate if we dose substantially higher than what we're doing here. We may see that the mechanism leads to a little bit of excessive contractions, so you can have a little bit of stiffness in the muscle. So that would cause a delayed relaxation. Let's say you squeeze your hand and you need to open your hand. That could be delayed, that relaxation, something that wears off as you repeatedly activate. It's a clinical phenomenon called myotonia. This is something that could happen. And also we did see that in our initial study in healthy participant when we dosed much higher than what we're dosing here. We did see that, which actually confirmed that we were having a compound that was finding the channel in also in human. But of course, we have not seen this. Now we have 32 patients with CMT that have participated, and we have not had any observations of this at this stage. Thomas, please chip in here. That is correct, no?
Yeah, absolutely. And what I can maybe also just say is that in the phase 1 study where we tested this in healthy participants, in general, outside of myotonia, we saw a similar frequency of adverse events in those that were treated with placebo as those that were treated with the active drug. So other than myotonia at very high dose levels, which Thomas just explained, we have not really seen anything so far.
And just to clarify, the drug that was used in the myasthenia gravis trial is the same drug used in the CMT trial.
Yep.
So I was asking about that. Oh, okay. So the role of CMT2 patients now, is that something that you're currently enrolling for?
Absolutely.
Yeah.
Both type 1s and type 2s are allowed into this study, regardless of the specific genotype.
And yes, for anyone asking, this is being recorded. It will be available on our YouTube channel in the by the end of this week. And when will the trial open for the third phase for more number of participants?
So we anticipate that this study will be finished and we will be able to conclude on this somewhere in the start of next year, maybe within the first half of next year. We hope to have it completed within that period of time. Then there are a number of, let's say, we need to learn from this study so we can optimize the design of the next study. Then we need, of course, to go through submissions to actually conduct the next study, and that has to be approved through the regulatory authorities and so on, and then we need to conduct it. So we would expect that we would be able to start the next study in somewhere early in 27, sorry, sort of start of 27, Q1, Q2, sort of that time in 27.
And for patients who've had surgery on their feet, which is pretty common with CMT, are they excluded from the trial?
Not per se. there will be an evaluation by the principal investigator and the site doctor to understand if the participants have any medical condition that will interfere with the study. And if that is considered by the investigator, then the participant cannot participate. But if not, then
Right. And we had a question here. Will trial participants be offered the option to continue the medication after the trial?
So that is a very, very good question. And I think at this stage, we do not have an open label extension to this particular study. And we are not, we're not able to bring that into this particular study. What we want to do with the current study is to make sure that we have designed it right and move as fast as possible to the larger study subsequent to that, which will be a longer study, which also will accelerate the path to actually getting this compound towards approval. But in the current study, there is really no further extension of the study medication. But it's a very important question. It's also very much on our mind. There is an aspect of this being also the fact that we are rather small company to some extent. I have to also admit that we are small. We are not the big pharmaceutical companies. We're very small, and we're doing the most we possibly can at this stage.
And back to the original scientific exploration of this drug, were you able to uncover any reasons why that nerve muscle communication erodes with CMT?
We have some reasons to believe that part of it is that the nerve ending over time deteriorates, and so you can see here the gray little circles between the nerve ending and the muscle fiber. These are small, let's say, vesicles of release of a substance called acetylcholine, or actually shortly abbreviated here as ACH. And what we definitely see in our animal models of CMT is that the amount of released substance becomes compromised and is reduced in CMT as a consequence of the motor nerve, let's say, having the disease. And if there's not enough released, then in fact the muscle fibers will not be activated. So by inhibiting CLT1, we can get basically larger effect out of the release acetylcholine. We're basically boosting the signal that is there,
Right.
and that becomes enough.
So it's not necessarily rescuing the motor neuron. It's just
over time we're seeing some really interesting aspects in our animal models. That over time we're actually seeing that if we can bring back the function, over time we're seeing a better and better connection happening over time here at the nerve ending.
Yeah.
So this is kind of the second stage effect of this MOA. There is kind of an immediate effect, which speaks more to recovery of muscle strength and function, but over time we're seeing a secondary sort of benefit of it in animals. So I think we just we have to put that caveat, of course.
Absolutely. And with CMT being a slowly progressive disease in most cases, can you share a little bit more about why a 21-day trial?
Yes, we can. And just to be, the way this was started out was that when you are developing drugs or a compound like this, you have to do parallel activities in the clinic, and then so also in different animal species, and making sure that you have the safety to cover your duration of the clinical trials. When we started this study, we had sufficient coverage from our toxicology studies to cover 21 days. We now have a lot more than that, but when we started it, we actually had the 21 days of coverage to do this. And I'll just point out again that this mechanism is very rapid. In fact, our myasthenia study, where we did see efficacy, which is actually shown here on the right, this Science translational paper, we only administered a single tablet, and we were able to see efficacy. So it works really fast. So 21 days should be sufficient to give us a clear understanding of which endpoints to move forward with in the future. And then by doing a short study like that, we are very well educated on how we can do the bigger study subsequently to get as fast as possible to the market.
That's very exciting. Usually, you know, trials will take 18 months, 4 months, and it can be very grueling on the patients and of course on the so to have something that could potentially show benefits that quickly is definitely very excited. How long do you think the effects last with per dose? Is this something that patients will have to stay on continuously for the rest of their lives, or how do you see that playing out?
This is how we see it. We see that it will be a tablet that one will take in the morning and then maybe in the middle of the day or sort of towards the early afternoon. And by having that with you, you actually have additional muscle strength and function to go through the day with a, let's say, excess or larger muscle strength, muscle capacity. But we do think it is a chronic therapy, and that speaks to the way that it works.
And for the phase 3 trial, do you still see the same age cap of 70 years old?
70 years old. I don't know that we have made a firm sort of position on that yet. So it's a little bit too early to say. We need to have the phase 2 data to really qualify an answer to that.
Right. Okay. I think we are just about done with questions. Does anyone else have any other questions? Feel free to raise your hand as well. I want to just give one last opportunity in case we haven't answered your questions. Courtney was kind enough to drop a link in the chat as well. HNF does have our own form to be connected with a research site coordinator for the clinical trials. So it's there in the link at our Donor Perfect link there for anyone who, again, would like to reach out to any of the trials to learn if they are a potential match. And we had another question here. Are you anticipating a huge placebo effect on the 6-MWT due to heterogeneity of affected population and large range of age?
Well, I think that's one of the benefits that we have of a short study. We will actually be able to do a pre-assessment and a post-assessment. So the heterogeneity in that respect, you actually are comparing to how you performed without to how you perform with. So that takes out a little bit the heterogeneity. Are we expecting any placebo effect? I think we have learned from many other clinical trials in many other indications that placebo effects are real things, and that's why we, of course, are including them to really make sure we have a true benefit of the therapy. But I would say the way that we conduct the study and the fact that it's a rather short study is actually helping us on this point.
And the trial, is it a double-blind trial?
Yeah.
And I think we had a question here for Oliver, if you're still available. Oliver?
Yes, I'm here.
Hi.
Can you hear? Hi.
Yeah.
I put it also in the Q&A now. My home country is Germany, and my son is also CMT1 patient, and he is in California. Both places are not on your list. Can we go to another place, to another country, maybe in Europe here, or here to another state in the US, and do it there?
Potentially, yes. The best thing to do is to contact the trial site nearest to you, and then discuss with the trial site if they can offer travel support. We do offer travel support, but it's not all participating sites that can actually accommodate this. And also, some sites they have a rather large population of CMT participants themselves. So it cannot be ruled out that some sites will not be able to accommodate foreigners into this study.
I understand. Would it be possible to just contact you?
Mhm.
Absolutely.
Yes.
Okay.
Absolutely.
Yes.
My email address is here if you want to write to me directly, and then we can try to figure out something.
Please,
I have it
please do,
already.
Oliver. Please do contact us.
Thank
so
you.
we can. Yeah.
Yeah, we know that our patient community is very excited to participate in any trial because there has been such a lack of clinical trials for CMT for so long. And so we see all the time patients are willing to travel outside of the country, outside of the state, disrupt their lives for an opportunity to be part of an exciting discovery like this that can benefit not only themselves, but family members. And because it is a genetic disorder, that it's not just, one family member, it's children, grandchildren, and even if it won't benefit them, they still want to create those opportunities for the younger generations to have a chance at seeing a treatment. So we appreciate that, Oliver, and
I
I'll
think, can I just address one point on that is, of course, we are conducting these clinical trials at the moment. We have not decided on countries for the subsequent trial yet, so there may also be an important option there in the future. And of course, eventually, when we are going to hopefully get this approach also marketed, we certainly aim to have it available as many places as we possibly can. So there are some steps towards this, but eventually, of course, it's going to be, for instance, in Oliver's case, of course, we're gonna we'll make it available in Germany. That, of course, will be our ambition to do.
Yeah. And Thomas, you'll be joining us in a few weeks at the CMT Summit and retreat that HNF is hosting in Nashville. You'll actually be our keynote speaker along with a few of other members of the NMD Pharma team. And we're really going to be diving deep, especially on day one, which is research day, into the science and how we can optimize and de-risk clinical trials such as yours, whether that's with digital health technology, designing clinical trials in a way that involves CMT community and feedback, and really including the experience of the CMT patient into the entire trial experience. So we're very excited to have you and to be hosting this event. We're pretty much almost sold out. So if you've been holding off on registering, we highly recommend you do that. We're almost at max capacity, which is very exciting. So there'll be a lot of wonderful conversations, and the second day is all going to be focused on patient wellness and adaptive mobility aids and all of the things that really affect us on a day-to-day. So we're really hoping this will be a very holistic experience for everyone attending. And thank you to both of you and to NMD Pharma and all of the incredible groundbreaking work and science that you have managed to achieve in a pretty short amount of time as far as clinical trials go. So thank you.
Well, just two things. We are very much looking forward to the summit. We're very much looking forward to learning, because this is a collaboration in that sense that we need to learn how we can optimize as well for the clinical trials and so on, and for the future. And thanks everyone for joining, and thanks for enabling this presentation today.
Yes, thank you so much. And if anyone has any questions, please reach out to Thomas at that email there. Also, you can complete the form in the chat. We'll leave the chat open for another minute or so. It's the HNF chat with the Donor Perfect link that will bring you to a form that you could fill out, and we can help connect you to your closest clinical trial site and answer any questions you have. So thanks again, everyone. Hope to see you at the summit, and we'll see you soon.
Okay. All right.
Thank you so
Thank
much.
you.
Bye-bye.
Bye.